This demonstration video will take you through the steps to successfully perform SDS Page polyacrylamide gel electrophoresis. Pre-made gels are available commercially, or if you choose to make your own gel, follow these next steps. Align the two glass plates such that the larger one is in back and clamp it into place in the casting stand. Make up your resolving gel. Briskly but carefully pipette the freshly prepared resolving gel into the space between the two glass plates, filling it to about 1 centimeter below the top of the plate holder. Pipette in 1 milliliter of water or butanol above the top of the gel and allow it to polymerize for about 1 hour. Once the gel is polymerized, pour off the butanol or water. Rinse briefly with distilled water. Make up your stacking gel. Then pipette in about 2 milliliters to fill most of the remaining space between the glass plates. Immediately, insert gently a comb of the appropriate thickness and well number. Allow the stacking gel to polymerize for about 30 to 45 minutes. Meanwhile, thaw and prepare your protein samples. Your goal should be to run equivalent amounts of protein in each well, aiming for about 10 to 20 micrograms of total protein per well. Calculate out the needed volume for each sample based on your Bradford assay results. You will end up loading the same total volume into your wells, so make up differences in protein volume by adding water to your sample tubes. Finally, add a reducing loading buffer to each tube. This will contain a reducing reagent like beta-mercaptoethanol. Place all of your sample tubes at 95 degrees Celsius for about 5 minutes. Disassemble your cast gel from the casting apparatus. Remove the comb and rinse the wells out with distilled water. Slide the gel into the electrophoresis tank with the larger plate facing outward and fill it with running buffer. Load each of your samples into separate wells and load protein standards into one well to serve as a ladder to identify the size of the protein bands that you will visualize later. Secure the top of the tank. Plug the electrodes into the power supply and run your gel until the samples have moved through the gel sufficiently. When your gel has finished running, you should see that the dye front has moved to the bottom of your gel. Turn off the power supply, remove the gel from the electrophoresis tank,